Have you ever noticed that humans act completely differently when they're in groups versus when they're just alone? But what you might not know is that cancer or tumor cells actually behave similarly, where they act differently when they're alone versus when they're in groups or clusters. Cancer is the uncontrolled growth of cells or a cell in the body that divide and divide even when they're not supposed to. But when it really becomes dangerous is when they divide and they enter the circulation and spread to different parts of the body, in a process known as metastasis.
Oftentimes they invade nearby tissue, maybe even gain access to the circulatory system, and in doing so have the capacity to move from one place to another.
Recently, in Dr. Cheng's lab at the North Carolina State University College of Veterinary Medicine, we've developed a unique method to watch tumor cells that are inside the circulation or the bloodstream and then see in real time how they're moving throughout the blood and then what mechanism they're using to exit once they're inside of the bloodstream. In this video, the tumor cells are labeled fluorescently blue and the blood vessels are red. Using a special type of microscope, we're able to look at these cells in real time and see how they're traveling through the blood vessels, and then more specifically, how when they're inside of the blood vessel, how they're exiting or getting out of the blood vessel and then forming more or new tumors. What we found is that the tumor cells exhibit a method of escape similar to stem cells, where they travel through the circulation and then they attach to the inside of the blood vessel wall and undergo a process known as angiopolosis, where the blood vessel wall restructures itself to push the cell from the inside into the surrounding tissue.
So angiopolosis was first discovered with stem cells, and we recently also found that circulating tumor cells also possess the same ability to exit the vessels using this mechanism.
And one interesting thing that we found out is that tumor cells that traveled as groups or clusters could also utilize this method to exit while maintaining their group or cluster adherence to one another. This is important because it's well known that tumor cells that travel and metastasize as clusters are much more likely to form secondary tumors or new tumors throughout the body. But the specific method or mechanism that they use to exit the circulation was unknown. We also observed that tumor cells were able to individually exit the circulation alone even after traveling as a group or a cluster. But next we want to determine exactly how the importance of them being a cluster versus a single cell affected their growth after they exited. So what we did is we looked at the tumor cells following their exiting of the blood vessel for 4 days and watched the rate of growth or proliferation. And what we saw is that tumor cells that exited as clusters had an increased ability to form tumors once they exited, versus tumors that exited as individuals or single cells, which tended to remain dormant and not show any growth over the course of several days. In summary, what we discovered is that tumor cells possess the ability to exit as both clusters and individuals through angiopolosis, but the tumor cells that exit as clusters through angiopolosis exhibit an increased ability to form secondary tumors, and these are the tumors that are accounting for the majority of new tumor formation that we see in cancer patients. This collectively is known as the cancer exodus hypothesis, and a better understanding of this will lead to better treatments for the prevention of metastasis.